These are the answers for the worksheet for chapter 6. It is actually chapter 6 and 7. This is the chapters that deal with the behavior of proteins, more specifically enzymes. We also studied their mechanisms and how they are controlled. The first exercise that we're going to do, I'm just going to zoom in a little bit more, is a match of the following terms and definitions. So, as you can see, we have terms on the left side and we have the definitions on the right side. We're going to start with apoenzyme. Apoenzyme, the definition is letter E, is a protein that forms an active enzyme system by combining with a coenzyme. It actually can do that also if we have a cofactor. The next term is going to be the active site, which the correct letter is going to be F. And as you can see, an active site is going to be the region on the surface of enzymes that fits the substrate. Term number 3 is activation energy, which the correct letter is A. That's the amount of energy required for a chemical reaction to occur. Some of you in other courses have heard that activation energy is the energy that has to be overcome by reactants to get to product. Both definitions are correct. Term number 4 is Km, and the answer is G, which is the value of substrate concentration at half maximal velocity. Term number 5 is substrate, which the correct answer is H. That is the substance enzymes act upon. Number 6 is an enzyme. An enzyme is going to be letter B, which these are proteins. So if you want to be more specific, it's the plural. So in terms of enzymes, these are proteins that speed up chemical reactions. Number 7, the term is zymogen or zymogens for plural, and a zymogen is going to be a letter D, okay, which is an active precursor or precursors for an enzyme that requires a biochemical change in order to become an active enzyme. And the last term is going to be holoenzyme, which the answer is C. And a holoenzyme is a catalytically active enzyme that contains a cofactor. Okay, so let's move on to part B. On part B in this worksheet, you guys have to classify the following reactions. I'm just going to zoom in a little bit so we can see all the different enzymes, all the different reactions. And when it comes to this, remember that there are 6 types of enzymes that you guys have to be familiar with. In this example, and for the exam, I also understand that I'll be giving you one or more substrates and the product corresponding to the change that that substrate is going to be undergoing, and that's how you're going to determine the class of enzyme that accomplished that reaction. So the different types of enzymes that you guys need to be familiar with include oxidoreductase, transferases or a transferase enzyme, lyase, isomerase. So I'm just writing them in singular. A ligase, and the last one is hydrolase. So let's start with problem A in part B. In problem A, looking at both substrate and product, you should, by looking at them and counting the number of carbons, oxygens, and hydrogens present in the molecule, you can see that they have the same chemical formula. The only difference between them is that the substrate is an aldehyde and the product is a ketone. And then whenever we have aldehydes and ketones that have the same chemical formula, they are isomers of each other. So the enzyme that catalyzes this reaction is an isomerase. Problem B in part B. As you can see, as the functional group in the substrate, we have a phenol. And what we have in the product is going to be a ketone, specifically a quinone. Understand that whenever we increase the number of bonds between carbon and oxygen, that implies that an oxidation reaction has occurred. So this is an example of an enzyme like an oxidoreductase accomplishing the product. On problem C on part B, we have two substrates and two products. So from biology, I hope that you guys remember that ADP is adenosine diphosphate. So the D stands for di. And in the product, we have ATP, which is adenosine triphosphate. So the T stands for tri. So the difference between the bigger molecule, the one that contains the carboxylate followed by the carbon that is double bonded to another carbon, single bonded to an oxygen that contains a phosphate, and the one that we have in the product that, as you can see, is a carboxylate bonded to an acetyl group, is that the phosphate went somewhere. Where did it go? Well, it went from the ADP to the ATP. So ADP accepted that phosphate. So here what we have is that an enzyme transferred the phosphate from one substrate to the next to then give you two products. So in this case, the enzyme that catalyzes C is going to be a transferase. On problem D on part B, here we have an amino acid. We have serine that gets dehydrated, and then the carbon that used to contain the OH, now it turned into a carbon that is double bonded to the alpha carbon in the amino acid. So in this case, because we are removing atoms and we're forming double bonds, this must have been accomplished by a lyase. And in the last problem, we have as a substrate the amino acid that is called homocysteine, and homocysteine is a derivative of the amino acid cysteine. As you can see, the difference is that there's an extra CH2, and when homocysteine undergoes this reaction along with the N5 methyl THF, we form methionine. So what you guys should notice here that there is a key feature of the THF that is missing. And as you can see, we have the N5 methyl THF. And the difference between the homocysteine and methionine is the addition of a CH3, which that group is called a methyl. So the enzyme that accomplished this transferred the methyl group from THF onto the homocysteine. So the enzyme that accomplished this had to be a transferase. Let's move on to part C. As you can see in part C, you have, well, it reads, triose phosphate isomerase catalyzes the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, and that can be illustrated, as you can see, in figure A. Figure B, which is below A, illustrates the chemistry at the active site of the enzyme. You have to determine the enzyme catalysis mechanism that this isomerase uses, and you also have to explain in detail how the amino acids glutamate 165 is missing the 1, so it's glutamate 165, and histidine 95 participate in the catalysis of the substrate when it is converted to product. So let's answer the first question. What type of enzymatic catalysis mechanism this undergoes. So when it comes to this, you have three options. It can be acid-base catalysis, it can be covalent catalysis, or it could be metal ion. There's other mechanisms out there, but those are the only three that you guys are responsible for. In order to get clues on which mechanism is happening, okay, then you look at part B. In part B, if you look closely at the three steps of that are occurring at the active site, you will see that there is proton transfers happening. So what that means is that somebody's acting as an acid, somebody's acting as a base. So that's why the mechanism, the enzymatic mechanism, is general acid base catalysis. Okay, then we have to explain it in detail. So I'm just going to move the page down, just zoom in a little bit closer to figure B so you guys can see how we can explain specifically what glutamate or glutamic acid 165 and histidine 95 are involved in this isomerization. So we're going to divide it into the three steps, starting with step 1. Okay, instead of illustrating things with arrows, how is typically done in organic chemistry, we are describing in words the arrows that we see that are present in figure B. Okay, so looking at figure B, you can see that glutamic acid 165 is first getting a proton from our substrate. So 165 is accepting a proton from dihydroxyacetone phosphate So that means that this residue at 165 is acting as a base. Okay? During the same process, as you can see in this mechanism, we have the carbon atoms that are part of dihydroxyacetone phosphate labeled. Okay? So in that first step, we have another proton transfer. Specifically, the oxygen atom at carbon number 2 in the substrate accepts a proton from histidine 95. So histidine 95 is acting as an acid. And that's pretty much what is happening in step 1. Let's move on to step 2. In step 2, one of the things that you guys should observe is that even though they're not labeled, this is still glutamate 165 to the left, and this is still histidine 95 to the right. The residues haven't changed. Now, in step 2, you can see that glutamate 165 is not doing anything, okay? The main exchange is going to be between the intermediate, that is called an indiol intermediate, and that histidine 65. So, to describe what is occurring in step 2, you can say histidine 95 is accepting a proton from the indiol intermediate which means that histidine 95 is acting as a base. Okay? You can also refer that the enediol intermediate is acting as an acid. Now, let's move on to step 3, the last step in this isomerization. In step 3, as you can see, we still have to the left the glutamate 165 or glutamic acid, and we still has the histidine 95. So when it comes to this, now you can observe that the only protonation deprotonation is occurring at the level of the new intermediate that is formed and the glutamic acid or the glutamate at 165. So we can specify that glue 165 is acting as an acid and donating a proton to the intermediate formed in step 2 to produce the final product.